Okay, we are back. So, we've covered the G protein coupled receptors in the last section, and now let's go on, and we're going to work toward the toll like receptors in this section. Okay, so in general, what are some other ways that we can activate these white blood cells, these macrophages and neutrophils, or how do they work? So, the idea is that when there are macrophages and they are successfully eating microbes and digesting them, as part of this, they're going to send out a couple of signals. And these are both little proteins that have slightly different names, but they're actually fairly similar. They're in sort of function. They're going to be called cytokines or chemokines, which are just two different families of small proteins that can bind to other immune cells and basically tell them to come in and check out whatever's going on here. So yeah, macrophages that are activated and have encountered some danger are going to release these things, and that's going to bring other cells in. So this causes the cells that line a blood vessel. So this empty thing in the center is going to be a blood vessel, something like a vein, for example. And it's going to cause those cells to increase production of receptors. And those receptors are what's going to snag some of these white blood cells. So the cells that are lining what's called the vascular endothelium, the inside of your veins and arteries, are able to tell parts of the immune system, hey, we could use some help. And they're able to capture things that will then come in. And so this process is called extravasation, which means going out of the vein. Yeah. And so basically they're going to squeeze through the space between these cells. I've seen some videos where they will actually, instead of squeezing through the edge, sort of the gap between them, which they loosen up, they'll sometimes actually just punch a hole right through the middle of the cells. And as long as they do it in the right way and go kind of thin as they go through, the cell can actually just reform and will be fine on the other side. But yeah, this is leakage of the veins and arteries. This is leakage of the vascular system. And if cells can get through, that means that a little bit of water can get through as well. And so this is where one of those signs of inflammation is going to come from, where you get a bit of swelling and redness. This is caused by water that has leaked out of your vascular system and is now kind of inside of your tissues. And so as a process, part of the process of the cells getting out, so these are the big macrophages, these are the little neutrophils, this will happen. And so the next thing that happens here is that we will actually clot the blood. So you saw on a previous slide we were mentioning those elastase-like proteins. Those are involved in starting the blood clotting process. So things that are immune triggers are also triggers for clotting, which kind of makes sense. When you get some kind of cut or some kind of damage, then you could have bacteria that are inside, which is very bad. And at the same time, you would need to patch up the hole. The bloodstream should be essentially sterile all the time. This is the idea. And that's what your immune system is trying to do for you. And so, yeah, it makes perfect sense that clotting and activation of white blood cells should go hand in hand. Okay, how does this work? Well, oh my gosh, yeah. So, remember, the versions of macrophages that you find in the bloodstream are called monocytes. These are the unactivated, sort of pre-macrophages, if you like. And after they bind the right receptors, and we will get to some of these adhesion molecules in later chapters, then they can actually, first of all, find a way to squeeze between these cells, just like that. And also, once they're on the inside, they will be activated. And so they will now go through some changes in the kinds of genes they express, and they will become actually, literally, macrophages. So yeah, that's the idea. And they're going to do this in response to chemokines. The chemokines are going to make the cells put out the receptors. The receptors are going to grab onto the monocytes. And once the monocyte's been grabbed, it's just waiting for a chance to burrow through some cells. And so that's the next thing that happens. There we go. And once it's through, it's going to cause more cytokines to be activated if there is more danger. But of course, for each cytokine activation, first you have to have something that a macrophage has picked up. And so when macrophages start showing up at the site and all the danger has been cleared, they then wouldn't get activated, and you would eventually stop making these cytokines and chemokines. All right, so the next proteins we're going to talk about are going to be toll-like receptors. And if they're toll-like, what is toll? I guess that's the question. Toll is a protein from a fruit fly. So this is a Drosophila fruit fly. And the way they discovered what this protein did was that when you knock out the protein, this is what happens to them. So here's our little fruit fly. That's its eyeball looking perfectly normal. Those are its little legs back there. And look at all this furry stuff, kind of like a lion's mane. That is all little strands of fungus that are going nuts and basically digesting this fruit fly while it is alive. And that's what happens when you knock out toll. It's a thing called the toll, and it is a receptor, so you call it a toll receptor in the fruit fly. It is basically unable to fight off fungus just from the world that would normally be controlled, so that it didn't do this to the fruit fly. They get this terrible, nasty fungal infection. And it turns out that toll is one of the very important genes that fruit flies have as part of their immune system. And it turns out that humans have not one, like toll, but 10 distinct copies of receptors that look like toll. They're basically the same gene, we just happen to have copied it 9 more times, so we have 10 total copies in our genome. And there are other creatures, like mice, that will, mice lack toll-like receptor number 10 that you find in humans, but if I remember right, I think they have toll-like receptors 11, 12, and 13 as well. So each of these toll-like receptors is able to bind to probably not just one thing, but a range of different generic markers of something that doesn't belong in the body. So there's something about the original toll that is actually binding to something that is generic to fungus. And that's the way that toll works. And each of our toll-like receptors binds a different range of things. It gives us a little bit of immunity in a kind of a non-specific way. So let's look at these things and let's talk about these things because they're good and worth knowing. And if you're going on in graduate school in this area or PA school or some kind of upper level nursing, like a nurse practitioner, or medical school or pharmacy school, you're going to have to learn this stuff. You're going to have a first year immunology that's probably going to use this same textbook. And so let's get these things, under your under your cap so that you can start to work with them. All right, the first one. So we're going to memorize all of these things. We don't have to memorize everything about them, but we need to memorize some things about them. So for each of these toll-like receptors, and we're only going to do 1 through 10 because these are the ones that we have in humans. We know a lot about the mouse proteins because mice are a wonderful experimental model, but we're not mice and we don't have these things, so and essentially why learn about something you can't use, you know? Interesting just to see what they do. Okay, so we're going to need to know at least one ligand for each toll-like receptor, and we're going to need to know at least one kind of cell that contains these toll-like receptors normally. And for each of these cell types, like monocytes, dendritic cells, mast cells, there are subtypes of all of these immune cells. And most of the subtypes may contain one of these things. There's probably a specific subtype for each one of these types of cells that does not contain the particular toll-like receptor. So make sure you take that with a grain of salt. But these are pretty good guidelines, pretty good general guidelines. Okay, so let's look at them. And this is what we're covering in this particular section. This and one other thing that's going to be coming up here in a bit, which is, boom, this table. So this table plus this table, which are really just an extension of the same thing. That's what we want to memorize. Okay, so toll-like receptor 1 and 2 are, we think, very primitive toll-like receptors. So if you go back in other animals, You can find toll-like receptors in pretty much any animal that is out there. There are a few specific lineages that may have lost these, but there's also another thing that works like a toll-like receptor called a nod-like receptor, N-O-D, nod, like nodding your head. And those work too. Yeah. And they do a similar job, so you can get by with one or another. So humans have these 10 toll-like receptors. And weirdly, we only know what 9 of the toll-like receptors do. And even for some of these toll-like receptors, I think we don't know everything that they do. So something like toll-like receptor 8, yes, you can set it off with single-stranded RNA, like viruses would make, but there are also probably going to be other ways to set some of these toll-like receptors off that we just don't know yet. Toll-like receptor 10 is actually very similar to toll-like receptor 1 and 2 and 6, and you see 1, 2, and 6 like to pair up together. So toll-like receptors tend to work as a team. And if they're a team of 2 different toll-like receptors, then they will be called a heterodimer. And if they're a team of 2 of the same, then they'll be called a homodimer. Yeah. But dimer means 2, and hetero means different, and homo means same. So there we go. So what are some of the things that these bind? And so since TLR10 is built quite similarly to and seems to be related to TLRs 1, 2, and 6, we think that it probably is going to bind something similar to one of the things that these bind, but nobody has a real good bead on exactly what it is that TLR10 binds right now. The best things that I've seen in the literature are indicating that it's some kind of bacterial factor, like a bacterial protein most likely, but even then, yeah, it's just there's not enough information. So you can just put unknown for that one. That's going to be fine. TLR1 is going to function with TLR2. So 1 and 2 are going to go together, and or 2 and 6 are going to go together. And these 2 heterodimers are going to bind one of these things. So a lipomannan is a sugar molecule attached to a lipid molecule, which is something that we don't do, but mycobacteria, things related to the bacterium that causes, tuberculosis. There we go. Those are going to make lipomannans. And so these are receptors that can pick that up. They can also pick up lipoproteins. So there are certain kinds of lipid molecule stuck to a protein that you would find in bacteria. And it's those kinds, the bacterial ones, that TLR 1, 2, and 6 are able to bind in their various combinations, 1, 2, and 2, 6. Lipoteichoic acids are what you would find. This is another lipid molecule attached to some other molecules. You would find it on the surface of gram-positive bacteria like Staphylococcus and Streptococcus, if I remember correctly. And cell wall factors, cell wall beta-glucan. So this is some of the sugar molecules that are attaching the cell wall of bacteria and fungus together. And so these are also bound. And so, yeah, if you have any of these things there, these are little pieces that would fall off of bacteria. And so this tells you that there are bacteria in the neighborhood, and so we need to do something about these bacteria. So zymosan is another marker, and that's going to be found very broadly on fungus. And so pick out some of these things. Maybe pick out a different one for TLR 1 and 2 and a different one for TLR 2 and 6. They all bind to all of these things at some level. So yeah, just pick me out one ligand for each of those. Pick me out a different one. Why not? Or remember more than one ligand. That's useful. Then you know a little bit more. Okay, TLR3, yeah, is going to bind double-stranded RNA. So some viruses like coronavirus, for example, don't have a DNA genome. They have an RNA genome. So they just have RNA. They don't have DNA. And when they copy themselves, they're going to take that one copy of RNA and they're going to make another copy of RNA that's kind of attached to it. That's just replication. And so this is a thing that happens when you have a virus around and generally does not happen except when a virus is around. And so TLR3 is good for detecting double-stranded RNA viruses. And yeah, that's good. Poly-IC I is inosine and C is cytosine. So these are 2 nucleotides. And you can stick them together, and honestly, we're not sure exactly why this works. I'm not sure what this is meant to be mimicking from nature, but this is another way to definitely set off TLR3. So this is something that immunologists figured out in the laboratory for when they wanted to study TLR3. And so TLR 1, 2, and 6, you can see these things are on, for example, let's pick out monocytes, because that's one of the things that's different. And these guys like eosinophils, basophils. So these are just very reactive cells. Macrophages are actually a step more complex. There are different subsets of them, and they're a little more regulated. And so TLR3 is a good way to get your macrophages fired up. And that is one of the things that we do with it in the laboratory. Okay, TLR4, and you can forget about CD14 and MD2, but these are part of the same complex. But let's just focus on the toll-like receptor. So this is going to bind to lipopolysaccharide, which is a lipid molecule attached to some sugar molecules. And it'll also bind to lipoteichoic acid, which is a different kind of lipid molecule attached to a different kind of sugar molecule that you're going to find on things like Staphylococcus, for example. And so, yeah, TLR4 is another one that we are very interested in setting this off sometimes. It's very useful for study. And so this is another macrophage trigger, if you like. Let's see, TLR5 is going to be set off by flagellin. So flagellin is the protein that makes up a bacterial flagellum, which is kind of like a tail that the bacterium is going to whip around in a circle, and it works like a propeller. It can either pull the bacterium along or push the bacterium along, depending on which way it is rotating. And some bacteria will have big bundles of like 5 or 10 of these flagella, and they'll kind of bundle them together like you can do with dreads and just have like a giant propeller that spins at the back. And so I guess that makes them go really fast. And so TLR5, yeah, that's going to be found in a variety of places, but also going to be expressed on the intestinal epithelium. So when you get bacteria in your gut that are starting to move around, and then, yeah, TLR5 is going to be on the job, recognizing that and calling in some cells to kind of deal with this, or make some antimicrobial peptides to deal with it. So TLR6 we already covered. It's going to pair up with TLR2 up here, and it's going to bind one of these bacterial markers. So if we go to TLR7 and 8, it's nice because they both bind to the same thing, but they're just found on different cell subsets. So TLR7 is going to bind single-stranded RNA, as is TLR8. And these are things that you would expect to find, like single-stranded RNA, there's lots of it inside of a living cell. But if you ever find single-stranded RNA floating past on the outside of a cell, that means that something bad has happened. So something has killed the cell and made it explode. And or you've got a virus that's actually made with RNA inside of it, like a coronavirus. And this has come along and set off TLR7 or 8. So TLR7, let's go with the plasmacytoid dendritic cells. That's a good one. And so these are going to be some of those cells that are hanging out in tissues and are then going to report to the rest of the immune system what they have found. And TLR7 is going to be one of the big activators. So when you get a virus, that's one of the things that happens. TLR8 is going to be, let's get neutrophils in the game here, because we haven't done much so far to activate a neutrophil. So TLR8 is a good way to activate a neutrophil. Yeah. All right, and then TLR9 is going to be, set off by DNA. But DNA, again, coming like loose DNA. DNA is normally something that's inside the nucleus of a living cell and is totally protected, where a TLR cannot get to it. But if you have loose DNA going around, that means something bad has happened. It's just like a blood trail, basically. And so, naturally, TLR9 is suspicious, as it should be. And so DNA that is not methylated is going to be what this recognizes. So bacteria actually spend a lot of time methylating themselves, methylating their own DNA. And that just means attaching methyl groups. And the way they attach them is they find a C nucleotide next to a G nucleotide. The P in between just says there's a phosphate in between. but between two nucleotides that are connected, there's always a phosphate. So we say CPG, but we could just say CG nucleotide pair, and that already indicates that you've got a phosphate in the middle, really. And so, yeah, some of those CG pairs or CPG motifs, you can call them. Two nucleotides is kind of a small motif. That's like a word for a repeating sequence. So motif is MOTIF, and it just means a repeating pattern of nucleotides or amino acids. So yeah, bacteria and some kinds of DNA viruses, like a herpes virus or like a pox virus, for example, would have unmethylated DNA and could set this off. And so for these, I don't know, let's get one of the other cells. Let's bring in B cells here. So that is a way to activate a B cell and cause it to start producing its antibody and maturing. Basically, it's a sign of danger. And then TLR10, we know it's on a lot of different cell types. So dendritic cells, eosinophils, B cells, and basophils, we just don't know for sure what it binds. We think it's probably related to something that TLR 1, 2, and 6 bind because it seems to be similar. But you know what? People have tried all those things and they cannot show what TLR10 binds. And so without that, it is, it's just something that we can't say right now. Okay, what do these things look like? And we'll get to the other table and then we will end this section. So a toll-like receptor's got two domains. So your little domain at the top, this is actually going to capture whatever the molecule is. The domain at the bottom is called a TIR domain, TIR. And these are found on toll-like receptors and also on the interleukin-1 receptor. It'll also have a TIR domain at the bottom. And keep an eye on these TIR domains. What they do, their purpose is that they can stick together. So TIR domains will make our toll-like receptors pair up, just like they're doing here. Or these guys, I think, could pair up if they wanted to. And the proteins that bind to toll-like receptors, basically the adapters that connect them to the rest of the cell, are also going to involve TIR domains, TIR domains. So that's why they've made it so it's like a yin-yang kind of thing, where you could take this one, flip it upside down, and lock it right into there. The shape is an opposite. Okay, so TLR 2 and 6, TLR 2 and 1, TLR 5, it's probably going to be 5 and 5, TLR 4 and 4, and there is a protein that sits in TLR4 called MD2 that interacts with it and helps it to bind things. But yeah, this is a nice little chart that can maybe help you remember some of these things. So the diacyl lipopeptides means a peptide, a little protein, that has two lipid chains on it. So two fatty acid chains. That's the diacyl part up here at the top. And so that's going to be one of the things that TLR 2 and 6 can bind. TLR 2 and 1 can bind to triacyl lipopeptides. Flagellin, that's going to be the bacterial flagellum protein. Lipopolysaccharide for TLR4. These are going to be located on the outside of the cell. So this is another piece of data that we're putting in. So in addition to this chart, I want you to know whether it is inside or outside the cell. So you can be making a little chart of your own, maybe on a piece of paper, and copy down one of these things from here, or you can copy down the diacyl or triacyl lipopeptide. That is fine. That is what we're talking about here. Then I want to know, is it on the inside or outside of the cell? So that's fine. You've got 1, 2, 4, 5, and 6 on the outside, and you've got 3, 7, 8, and 9 on the inside. So the endosome, this is where we're digesting things. And so if we just burst something open, and it happens to have double-stranded RNA, single-stranded RNA, or DNA inside of it that is not methylated, then boom, that's going to set off alarm bells. That just means you ate something that is either alive or a virus. And so that's how a macrophage knows that, whoa, yeah, we got one. This is the little tug on the line like you would have if you're out fishing that kind of indicates to the toll-like receptor that this is a sign of danger. Something bad is happening, and we need to figure out a way to deal with this. Okay, So, for example, TLR 1 and 2 will be monomers on their own. The tier domains would like to associate, but they're not really strong enough to associate. But when you get a lipopeptide, and so here is a triacyl lipopeptide with 3 fatty acid chains on it, then that can actually bind to both of these. It can lock them together enough that the 2 tier domains are now together. And so here's the structure of it. And look at this thing. Look at those awesome beta strands across the bottom. Look at these alpha helices on the top and just all these loops at the side. Those are some beautiful looking proteins. And then here is our TIR domain, the tier domain, in the bottom. And so this is the thing that's going to connect us to the rest of the inside of the cell and the rest of the signaling process. And so shown here, this is a lipopolysaccharide molecule in association with MD2, so that's the little protein, the little red thing that's kind of clamped in here. And CD14 is, I think, going to be the purple one, sort of bluish purple one over here. And yeah, this is 2 copies of TLR4 together, and they have bound to this lipopolysaccharide, which I believe is the orange thing up here. And so, yeah, when this comes along, then boom, 2 copies of TLR4 are going to get locked together. The 2 tier domains are going to bind to each other, and boom, we get signaling. All right, the last thing you need in your table, and we're going to leave off TLR 11, 12, and 13, because those are only found in mice, is we need to know what is it that connects our protein to the rest of the immune system. And so there are only a couple of choices. So for TLR 1 and 2, it's going to be MyD88. So that's how we say this. And that is a protein that has a tier domain in it. So the tier domain of MyD88 will bind to the tier domain of the toll-like receptor. And they'll stick together. So we don't have to remember, for the ones where it says MyD88 or MAL, let's just leave off the MAL. Let's just stick to MyD88. That's a little easier. And over here where it says TRIF or TRAM, TRAM is another TRIF-like protein, and so let's just remember TRIF. So for 1, 2, and 2, 6, yeah, it's just MyD88 that binds both of them. For TLR10, we don't know, but we could speculate that it's probably going to be MyD88 and or MAL that are going to bind to these. So, but since we don't know, we're not going to just wildly speculate, but I'm just saying, as you live in this world, eventually somebody's going to figure it out, and that's going to be the most likely answer. But for TLR3, we want to know that it's TRIF. For TLR4, we want to know that this can activate both pathways. So MyD88 pathway and the TRIF pathway. And so you don't have to remember MAL or TRAM, but you can remember them if you want. I'll probably throw you a little extra credit if you can get those on there. And then for pretty much everything else, 5, 7, 8, and 9, it's just going to be straight up MyD88. Doesn't seem to bind to MAL, and definitely not to TRIF or TRAM. So there we go. Those together make up our super table. So this is a super table that, again, you're putting together. It's going to have the toll-like receptor or combination of them. It's going to have the thing that the toll-like receptor binds. Remember to mark this as diacyl or triacyl. It's going to have at least one of these cells from the list that the toll-like receptor is found on. It's going to tell you whether the toll-like receptor is expressed on the outside or inside, of the cell. And it's going to tell you, the last thing, what the toll-like receptor signals through. And we're only doing toll-like receptors 1 through 10, and there are a couple of these categories where we don't know what 10 is doing. And so 10 is going to be unknown, and then nothing else on there. So the 10 column is like a gimme, but at least you know. You know not to put anything else in there. Okay, that's a good place for us to pause. Yeah, we'll come back to these MyD88 and what they do in the next section. So for now, get out there, make that little table, and you're going to learn that table. And again, I guarantee that you are going to have this question. Now, you may only have half of them, or you may have all of them. I don't know. I may just put this on everybody's test. But yeah, you'll at least have to know some of the toll-like receptors, and probably all of them. So let's just say all of them. So thanks very much.